Hi, general biology students. So we are in chapter 4, and this will be the last chapter before your first lecture exam. So your first lecture exam will be on chapters 1, 2, and 4. We've skipped chapter 3 for now, and we'll come back to it. Chapter 4 is a little lengthy, so feel free to pause this PowerPoint and come back to it or the lecture to listen to it if you get a little bored. I'm going to try to get through it in one take. Unless my kids interrupt me, otherwise I will do kind of part A and part B here. So chapter 4 is all about the tour of the cells. So this is a really good chapter to kind of give you a basis for what a cell looks like. We first need to talk a little bit about microscopes. Improved microscopes and techniques have vastly expanded our view of the cell. For example, in 1665, the man by the name of Hooke used a very crude microscope to examine a piece of bark from an oak tree, and he compared the structures he saw to little rooms, which in Latin means cellulae, and the term cell stuck. So that's where the cell word came from. A few decades later, a man by the name of Leeuwenhoek used more refined microscopes to view numerous subjects, including blood, sperm, and pond water. And in this chapter, we'll just explore the cellular basis all of life. So our knowledge of cells has grown vastly, and that's really due to the use of microscopes as well, and just the ability to see the cell at a more zoomed in version. So this is just a look at an example of a cell, and we're going to zoom in on different types of cells here. So chapter 4, big ideas. We'll introduce the cell, we'll look at the nucleus, ribosomes, the endomembrane system, energy converting organelles, which will be the mitochondria, and then the cytoskeleton, which is like the bones of the skull and the cell surfaces. So a light microscope can display living cells, and if we were having class in person, one of our labs would be using a light microscope to look at cells. The greater magnification, meaning more zooming in, and resolution of scanning and transmission electron microscopes reveal the ultrastructure of cells. The word magnification refers to the increase in an object's image size compared with its actual size. And the word resolution is a measure of the clarity of an image. So in other words, it's the ability of an instrument to show two nearby objects as separate. In the mid-1800s, studies of cells led to the cell theory, which states that all living things are comprised of cells and all cells come from other cells. So which type of microscope would you use to study the changes in shape of a living human white blood cell? Would be a light microscope. The finest detail of surface texture of a human hair we haven't really talked about, but we would use what we call a transmission electron microscope, which is just a more zoomed in version. And the detailed structure of an organelle in a liver cell, we would also use a scanning or transmission electron microscope. So here's a light microscope view, light micrograph of the unicellular organism, their paramecium. Also in the mid-1800s, excuse me, sorry, I went the wrong slide. Here's a scanning electron micrograph of the paramecium. So this is just a look at the differences between a light micrograph. So this is using a light microscope, which is what we have in lab. So this is how we would view the organism paramecium using the light microscopes that we have in lab. This is a scanning electron micrograph of the paramecium. So you can see the more in-depth zoomed in detail of the picture. We do not have these in lab, but it's the SEM microscope is another great scientific advance for viewing organisms on a cellular level. And this is the most zoomed in microscope, the transmission electron micrograph. So it sends out electrons through the specimen and they bounce back, and you're able to see even more detail within the specimen. So this is a transmission electron micrograph. So the most kind of zoomed in we can get, this is at 9100, almost 9200 times magnified of the organism toxoplasma. Here's the differential interference contrast micrograph of paramecium in the light microscope. So using kind of an interference technique to show some differences between structures within the organism. This is a great graph showing the size range of cells and related objects. So for example, around 1 to 2, kind of 3 meters, we have the human height. The length of some nerve and muscle cells is 1,000 millimeters. The chicken egg, the frog egg, the paramecium, and the human egg, these are all things we can see with the naked eye or unaided eye. So without a microscope, we can get down and see the paramecium and even the human egg or the ovule. But with the light microscope, we can see most plant and animal cells, the nucleus, most bacteria, and mitochondria. We'll talk about what those mean. With the electron microscope, whether that's a transmission or scanning electron microscope, then we can see even more detailed, more zoomed in. The smallest bacteria, viruses, ribosomes, which create proteins, lipids, small molecules. Atoms are kind of out the range of microscopes at all. So this is just a zoomed in version of the graph. So the things we can see with the unaided eye, so meaning without a microscope. And then a more kind of zoomed in version of the rest of the chart. Again, things we can see with the light microscope, which again is the type of microscope that we would have in lab and school. And then the electron microscope gives us a little more detail. Just another look at the chart, the size range of cells and related objects in a horizontal layout. So again, feel free to pause the lecture recording at any time to kind of take your own notes and look at this more in depth. The small size of cells relates to the need to exchange materials across the plasma membrane. So the microscopic size of most cells provides a very large surface to volume ratio. And when we talk about the surface of the cells, we're referring to the plasma membrane, which is a phospholipid bilayer with embedded proteins within it. Some of these proteins form channels or tunnels that will shield ions and other hydrophilic molecules, meaning water-loving molecules as they pass through the hydrophobic center of the membrane. Other proteins will serve as pumps, using energy to actively transport molecules into or out of the cell. To convince yourself that a small cell has a greater surface area relative to volume than a large cell, compare the surface to volume ratios of the large cube on the left and the smaller cubes on the right. So the total volume is the same between one large cube and the same size of about 27 smaller cubes making it up. The total surface to volume ratio is a lot smaller in the one large cube versus the many, many smaller tiny cubes. So the surface to volume ratio is about 6 times that with the many smaller cubes. So this is a look at the plasma membrane. So we call it a lipid bilayer, meaning there's 2 sets of these kind of gray hydrophilic heads on the top and the bottom, and then their tails are in yellow and they will point and touch each other because they are hydrophobic. So on the outside and inside of the cell is mostly water. So we have the water-loving heads touching the exterior and interior of the cell, and the hydrophobic tails, which are water-fearing, will touch each other. And then we have different proteins embedded throughout the plasma membrane. We can have a channel protein, hydrophilic region of a protein, and hydrophobic region of a protein. So this is the plasma membrane structure. It's a lipid bilayer because there's 2 layers of lipids present. All cells have a plasma membrane, DNA, ribosomes, and cytosol, which is all the fluid within the cell. Bacteria and archaea consist of prokaryotic cells, which do not have any membrane-bound organelles, and all other forms of life are placed in the domain eukarya and have eukaryotic cells. And eukaryotic cells are distinguished by having a membrane enclosed nucleus, and the nucleus is like the control center, and many membrane-enclosed organelles that perform a specific function. Prokaryotic cells will be much smaller and simpler in structure. So this is a look at a diagram on the left and an electron micrograph on the right of a typical prokaryotic cell. So you'll notice there's no membrane-bound nucleus. You do have this the structure called the nucleoid, which contains all the chromosomes. So normally in the nucleus, that's where all your genetic material lies. And in a prokaryotic cell, it's just kind of free-floating where these chromosomes are. It still has a plasma membrane and a cell wall. Bacteria could often have a flagella, which allows it to move. And this specific type of bacteria is called Helicobacter pylori, which can cause stomach ulcers. Not fun. So here's the rod-shaped bacterium. Again, you can see the flagellum that helps with movement, the capsule. And then again, this is a prokaryotic cell, so there is no membrane-bound nucleus. All the genetic material is kind of just free-floating within the cell. And then the transmission electron micrograph. So here's an actual drawing, and then the transmission electron micrograph, again, which is almost 21,000 times magnified, which is a crazy amount zoomed in, actually gives us a pretty good representation of what the artist drew. Eukaryotic cells are partitioned or separated into functional compartments, and these membrane-enclosed organelles will compartmentalize the cell's activities. The organelles and other structures of eukaryotic cells can be organized into 4 basic functional groups. The nucleus and ribosomes carry out the genetic control, so creating proteins, passing along genes and genetic information, DNA. Organelles involved in the manufacture, distribution, and breakdown of molecules include the endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, and peroxisomes. The mitochondria in cells and chloroplasts in plant cells function in all the energy processing, and all the structural support, movement, and communication between cells are what makes up the cytoskeleton, plasma membrane, and the plant cell wall. For you can have a checkpoint, ask yourself the structures in the plant cell that are not present in the animal cell. So here we have a look at a generalized animal cell, and that checkpoint question we'll kind of answer as we go through this PowerPoint. We'll talk about a typical animal cell. You can see the nucleus. It has its own nuclear envelope. The nucleolus, the chromatin is the condensed DNA. The rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, the plasma membrane, peroxisomes, the centrosome is a pair of centrioles that will help to separate out the cell during cell division. And then the cytoskeleton kind of makes up our bones of the cell. We have 3 types of molecules there. We have the microtubules, microfilaments, and intermediate filaments. Here's a generalized plant cell. So now you can see some of the differences here. A plant cell does have a nucleus, rough endoplasmic reticulum. You'll notice a lot of similar things. The big one that's different is we have a cell wall, and there's also a chloroplast, which will give this the plant cell its green color and will be where photosynthesis occurs. And there also usually exists this central vacuole space. So we'll start with the nucleus and ribosomes, which contains the cell's genetic instructions. The nucleus will house the cell's DNA, which directs protein synthesis via messenger RNA. And then we have subunits of ribosomes, which will be assembled in the nucleolus, describe the processes that occur in the nucleus, and we'll talk about that now. So here's a cross-section of the nucleus with a superimposed transmission electron microscope showing the other side. Here's just a standard cell. You can see the nucleus, the nuclear envelope, and then the nucleolus with the kind of condensed chromatin around it, which contains all the cell's genetic material or DNA. There are pores that exist in the nuclear envelope, and that will allow messenger RNA to take the transcribed genetic material out of the nucleus into the ribosomes where proteins will be made. Ribosomes are composed of ribosomal RNA and proteins, and they will make proteins according to directions that they receive from the DNA in the nucleus. Cells that make a lot of proteins will have a large number of ribosomes because ribosomes make proteins. Try to get that into your head. Ribosomes make proteins, so if you have a cell that requires a lot of proteins, it will most likely have a lot of ribosomes. So here's a look at the locations and structures of the ribosomes. The ribosomes can be bound or connected to endoplasmic reticulum, And if they're connected to endoplasmic reticulum, we call the endoplasmic reticulum rough because it's studded with all these ribosomes. And then ribosomes can also be free floating within the cell outside of the nucleus, and we'll call them free ribosomes. And this is the structural unit of a ribosome. It's made up of these two subunits where the messenger RNA, that's mRNA, will kind of pass through. And as it passes through, different processes occur that begin to make proteins that's seen coming out there. The endomembrane system refers to all the organelles kind of interconnected in what we call the endomembrane system in the eukaryotic cell. They will interact in the synthesis, distribution, storage, and export of molecules. So kind of think of these membranes as kind of like an Amazon distribution center. The endoplasmic reticulum is a biosynthetic workshop. It's a membranous network of tubes and sacs, smooth endoplasmic reticulum because it does not have ribosomes, so we call it smooth. This will synthesize lipids and process toxins. So you'll find a lot of smooth ER in your liver, which is a place where all drugs and alcohol go because smooth ER helps to process all that toxic material. Rough ER, which is called rough because it's studded with ribosomes. Because it has ribosomes, it'll be important for creating proteins. And why do we call the endoplasmic reticulum a biosynthetic workshop? Because it's synthesizing lipids, which are fats, or it's synthesizing proteins. So it's kind of this workshop. So here's a look at smooth and rough endoplasmic reticulum. Again, rough endoplasmic reticulum has the ribosomes and smooth does not. And they're often interconnected. And again, they are kind of these membranous sacs that will do a lot of the biosynthesis of materials within the cell. So here's a look at synthesis and packaging of secretory protein by the rough ER. So here we have rough endoplasmic reticulum. The bound ribosome is connected to it, so the protein is being made. It goes directly into this tubule system of the rough ER. This protein forms a sugar chain, and eventually the transport vesicle breaks off of the rough ER and takes that sugar chain inside of it to the correct location that is needed, whether it's inside the cell or outside the cell. And then the Golgi apparatus will modify, sort, and ship all the cell products. And the Golgi consists of stacks of sacs in which products of the ER, the endoplasmic reticulum, will be processed and then sent to other organelles to the cell surface. So here's your Golgi apparatus. It'll have a receiving end where it'll get vesicles from the endoplasmic reticulum. Then it'll process them, sort them, figure out where it needs to go. Then parts of it will be broken off into different transport vesicles from the Golgi. This is called the shipping side, and those transport vesicles will then be sent out to other locations. Then we'll talk about mitochondria and the structural support. Lysosomes will be the digestive compartment within the cell, and lysosomes will house enzymes that can break down ingested substances and anything that's damaged. So a lysosome is like a recycling center because it'll break an ingested substance or a damaged substance down into its appropriate components that can still go on and be used. So here's a look at a lysosome containing digestive enzymes. It'll eventually probably fuse with the plasma membrane to kind of spit out what's inside of it to get that waste to the exterior of the cell. Or it will take in food vacuoles, and it'll take in those food vacuoles and digest them before it gets further into the cell. Here's a vesicle containing damaged mitochondria where the lysosome will kind of merge to it and help digest the damaged structure. Here's a great animation talking about lysosome formation. So again, please download the PowerPoint and take a look at that animation. A vacuole is a large vesicle that has a variety of functions. Some protists have contractile vacuoles, and plant cells contain a large central vacuole that will store molecules and waste and facilitate growth. Is a food vacuole part of the endomembrane system? Not often we'll see food vacuoles in just plant cells. So here's the plant cell showing a contractile vacuole in the paramecium, which is a unicellular eukaryote. Here's a central vacuole in a plant cell, kind of this open space. It looks like there's nothing there, but it's a central vacuole. And here's another great video of a paramecium vacuole in that cell. The organelles of the endomembrane in review are interconnected structurally and functionally. We'll show a figure here that will summarize all the relationships among the major organelles of the endomembrane system. So here's a look again. We see the nucleus, the nuclear envelope with pores, where all that genetic material will pass through and be connected to ribosomes, which will be found on the rough ER. Those ribosomes, which will create proteins, which will travel through transport vesicles, which will head to the Golgi apparatus, which will sort all that, those proteins or information, and then eventually transport all the proteins out of the Golgi to transport vesicles either outside the cell or inside the cell. A lysosome will help digest anything damaged or anything that needs to be broken down. How do transport vesicles help tie together the endomembrane system? The transport vesicles really take everything that's being biosynthesized, whether that's a protein, anything else that is being made in the ribosomes. In the transport vesicles, will take it on to its next step or either transport it outside the cell. Peroxisomes then are metabolic compartments that do not originate from the endomembrane system. So peroxisomes kind of have their own function and help to break down and digest materials, but they're kind of their own organelle. They're not involved with that system. The energy converting organelles, then the big one we're going to talk about is mitochondria, which are organelles that carry out cellular down there. Cellular respiration is the process by which your cells take in glucose, which is basically from anything that you eat, any sort of food material. Cellular respiration takes glucose and turns it into ATP, which is the body's currency for energy. And this cellular respiration, taking glucose and turning it into energy, occurs in the mitochondria. And the mitochondria have two internal compartments to them. They have an intermembrane space, which is a narrow region between the inner and outer membranes. And then they have a mitochondrial matrix, which contain the mitochondrial DNA, ribosomes, and many enzymes that catalyze some of the reactions of cellular respiration. So here's a look at the structure of mitochondria, the outer membrane and the inner membrane. And between the two is this intermembrane space. And then within the intermembrane, we have what we call the mitochondrial matrix. And the mitochondrial matrix makes these folds called this. So we're going to talk a little bit about plant cells too. So chloroplasts will convert solar energy to chemical energy in a process known as photosynthesis, which is the conversion of light from the sun to the chemical energy of sugar molecules. So plants are able to take in carbon dioxide and water and create glucose. Chloroplasts are the photosynthesizing organelles of plants and algae. And this is a look at a chloroplast within a plant cell. Membrane in a chloroplast appears to be the most extensive. And you can kind of see here the parts of the chloroplasts are labeled, the inner and outer membranes, the stronum, the granum, and the diacolyloid. And you can see here both membranes are kind of about the same, and probably because they're both doing the equal amount of work. The endosymbiotic theory states that mitochondria and chloroplasts were formerly small prokaryotes that began living within larger cells. So again, this is one to mitochondria and chloroplasts having similar structure. Here's a look at the endosymbiotic origin of mitochondria and chloroplasts. taking through the different steps of development. Step 1, step 2, and step 3. And I'll let you guys kind of work through the lecture, which is awesome. I want you guys to really focus on the different organelles and what is the structure. Cytoskeleton and cell surfaces refers to kind of the cell's bones. So this includes microfilaments, intermediate filaments, and microtubules. And their functions include maintenance of cell shape, anchorage and movement of organelles, ambioid movement, and muscle contraction. Which component of the cytoskeleton is most important in holding the nucleus in place within an animal cell, guiding transport vesicles, and contracting muscle? And we'll answer those questions here. So here's a look at the 3 types of fibers of the cytoskeleton: microtubules, labeled with the green fluorescent molecules on the left, intermediate filaments labeled yellow-green in the center, and microfilaments labeled red. Microfilaments are actin and myosin, which will make up muscle contraction. Intermediate filaments are shown here, and then the microtubules are the largest. So here's the microtubules, the round strands of microtubules in green, and the enlarged view shows illustration depicting this structure. It's 25 nanometers wide, which is the largest of the microfilaments, and it's made up of small balls of tubulin protein coiled kind of like a phone cord. Here's a look at the intermediate filament, which shows the structural feature. It's 10 nanometers wide, so a little bit smaller, and it's made up of the fibrous point proteins coiled together. And then here is a microfilament. Examples of these are actin and myosin, so they're the thinnest, about 7 nanometers wide. Actin and myosin will work together to contract and slide past each other in muscle contraction. So here's a look at the microtubules and the intermediate filaments, and then the microfilaments and what they look like under a microscope. Scientists discovered the cytoskeleton using the tools of biochemistry and microscopy. In the 1940s, biochemists first isolated and identified the proteins actin and myosin, which form your muscle cells. In 1954, scientists, using these newly developed techniques of microscopy, really established how the filaments, actin and myosin, interact in muscle contraction. Next chapters identified actin filaments in all types of cells. Researchers were then able to tag actin proteins with a fluorescent molecule and inject them into a living cell. And this technique enabled scientists to visualize the dynamic behavior of the cytoskeleton proteins in living cells. So here's a look at a fluorescent micrograph of the cytoskeleton. Microtubules are in green and microfilaments are in reddish orange. Then we'll talk about cilia and flagella, which move when microtubules bend. Eukaryotic cilia and flagella are locomotor appendages, meaning they help in movement. They're arranged in a 9+2 arrangement. Flagella are longer than cilia, and they propel a cell by a whip-like motion. And the only human cell that has a flagella is the sperm cell. Cilia, in a little bit different way, will work more like a coordinated oar of a rowing team. And though they're different in length and beating patterns, cilia and flagella have a common structure and mechanism of movement. This is primary ciliary dyskinesia, also known as immotile cilia syndrome. It's a rare disease in which cilia and flagella are lacking motor proteins. PCD is characterized by recurrent respiratory tract infections and immotile sperm. And how would you explain these seemingly unrelated symptoms? So this disease affects cilia and flagella. I kind of mentioned it before. The human sperm is the only human cell that has a flagella, so it would attack the sperm cells, making them immotile. which would cause some reproductive issues most likely. The respiratory tract infections, so cilia are often found in your respiratory tract. So your trachea, your nose, your lungs, your cilia help to trap any harmful substances that you breathe in and then try to move them out. So this disease would affect both because they are made up of the same type of structure. So it could affect things in your respiratory tract as well as sperm cells. So here's a look at cilia cells lining the respiratory tract, again, about 5,000 times magnified. So this is a good microscope, a scanning electron microscope. And here's a look at a flagellum on a human sperm cell. Here's the internal structure of a flagellum or cilia, again, showing the arrangement kind of these 9 pairs of tubules that make this kind of arrangement, which help with the locomotion. The outer microtubule doublet, the central microtubules, and then the cross-linking proteins. And then we have motor proteins, the dyneins, which help to actually move. The internal structure, again, of the flagellum or cilium, the cross links between the outer and inner doublets, which will help with movement and locomotion. And here's a great animation showing cilia and flagella. Again, another video, download this PowerPoint from Canvas and watch these animations. They're really great. The extracellular matrix of animal cells will function in support and regulation. And we call extracellular matrix ECM. And when we talk about ECM, we mean anything that's not cells, so extracellular matrix. This will bind tissue cells together, support the plasma membrane, and communicate with the cytoskeleton within the cell. The ECM may attach to the cell through other glycoproteins that then bind to membrane proteins called integrins. So this is a look at extracellular fluids. So anything outside the cell, we see collagen fibers connecting glycoproteins connected to integrins, which will then connect to cytoskeleton. So we talk about cells and what makes up the cell inside and the border of the cell, but also in a tissue, if we talk about extracellular matrix, the things that surround the cell on the outside are also very important to supporting the cell and communicating with the external environment. Neighboring cells will often adhere, interact, and communicate through specialized junctions between them. Tight junctions will bind cells to form a leak-proof sheet. An anchoring junction will rivet cells into strong tissues. Gap junctions allow ions and small molecules to flow from cell to cell. A muscle tear injury would probably involve the rupture of which type of cell junction? And we'll talk about that. These are the 3 types of cell junctions: tight junctions, anchoring junctions, and gap junctions, just showing junctions between neighboring cells. Tight junctions prevent fluid from moving across the layer of cells. Ions are small molecules. Here's desmosomes, which are involved in tight junctions, gap junctions, and tight junctions. So feel free to watch all those animations as well. Cell walls then enclose and support plant cells. A plant cell, but not an animal cell, has a rigid cell wall, and that's one of the main differences. The cell wall protects and provides skeletal support that helps keep the plant upright on land and is primarily composed of cellulose. Plant cells have cell junctions called plasmodesmata that will allow plant tissues to share water, nourishment, and chemical messengers. So here's a look at plant cell wall. You can see here how it does kind of have junctions between the two to allow things to pass through them. The eukaryotic cell structures, again, can be grouped on the basis of the 4 functions: genetic control, manufacturing, distribution, and breakdown of materials, energy processing, and structural support, movement, and internal communication. How do mitochondria, smooth ER, and the cytoskeleton all contribute to the contraction of the muscle cell? And that will be something we talk about a little bit later. Mitochondria will help provide the energy for muscle contraction. Smooth ER provide the calcium, and the cytoskeleton provides the actin and myosin that will slide past each other to contract your muscles. Here's again another great look. This is an extremely small, so you might have to zoom in here when you download the PowerPoint, but it's a good review of the cell structures and what their process is. So a lot of the questions from this chapter I'm going to ask on cell structures, animal cell structures, and what their structure is and what their function is. Here's again a more look at the chart, a little bit zoomed in, so feel free to pause the lecture video, download the PowerPoint, and study these tables because these will be a really great source of review from this chapter. You should be able to now do these. So this is kind of the checkpoint part. There's a couple slides here. So feel free to work your way through kind of those review questions. Use the book to answer, help kind of go through their review questions. Use the review questions I posted. This is a look at metric measurement equivalence. We talked a little bit about this in our first lab, how many millimeters are in a meter, centimeters in a meter. So this takes you through some of the metric system equivalents. Here's a figure, again, reviewing concepts. These kind of last couple slides at the end of the PowerPoint review some of the key concepts that we talked about. Here's the paramecium drawing with the flagellum. Here's a great practice. If you can label all these structures, labeled I through L of the internal parts of the cell, that's also a great review slide. Here are the poles of a dividing cell. This is talking a little bit about mitosis, which we did not get to in this chapter yet. And that is the end of this chapter. So thanks for listening, guys. And this will take us to our first lecture exam, chapters 1, 2, and 4.